So we are going to try something to spend more time on some of the technical things and provide a little bit more time for discussion. Hopefully this will work, and we'll be looking forward to getting your feedback after this session on how we were able to improve. Another thing that we want to mention is that, of course, a lot of the materials are available on the educational website, on the online sessions. We do have recordings of the session. So we really see this hands-on session that we do today and every week as an opportunity for us to practically do some things and then to have a discussion and answer any questions. So we did provide the files for today's session, and I just want to make sure that everybody's seen them. It's on the discussion board. And like I said, we're going to start with going quickly through what we want to talk about, and then we will try to leave more time to go back, go through things slowly, and it will be more focused on what you feel is needed rather than us kind of directing the conversation, okay? So I will go through the presentation that we have for today, cover the major points, and then I will turn it over to Sahil, and Sahil will be able to go through the things a little bit slower, a little bit more in detail, and answer any questions. If he can't answer some of the questions, we will make sure that we record your questions, and then we will provide another session specifically addressing the questions that everybody had. Again, after this session, we'll try and have a conversation with you, and we'll try to get that feedback from you. So please fill out those forms. That helps us a lot to adjust our pace and to also address the things that everybody feels we need to. So let's start by going through where we are in the program. In the program, we are at our third meeting. The third meeting is focused today on filtering, removing noise, and applying some normalization techniques to next generation sequencing, the table of expression that we generated last time. And then how to summarize. What do we see in this table of expression? We have thousands of genes, thousands of numbers. How can we summarize some of the methods that we can summarize this information with? So I will kind of go through some of these things, and then Sahil will go back and make sure that individual components of what we talk about are clear. And if you have some questions, please do let us know. You can already put them in chat, and we'll make sure to address those. Now, just in terms of everybody's progress, like we said, we have this chart that lets you see where everybody is. So you can see on this chart, there are some educational points that you get for completing the online materials. There are some points that you get for doing the practical hands-on things. Here you can see on an individual level kind of who's doing what. So this would really help if you took a look at this every once in a while, kind of saw where everybody is, because at some point we want to transition to a project that some of you have expressed a desire to work on an independent project. And so having sufficient amount of points gets you access to the research license of the platform, and you can explore the different portions that you can use to do your independent project. Okay, so this is where we are now. And we did an in-depth review of the independent project ideas that everybody posted. And in the next session, Justin will be presenting some of those topics and some of the relevant project ideas that we already have. And we'll be able to set up groups and start talking about how to prepare for a project, what to do with those. Review. What did we do last time? Last time we talked about raw FASTQ files. So we addressed two separate issues. One issue of pre-processing, and that is more related to the way that these reads are generated to the sequencing process. And the second one, mapping. Okay, how do we map the short reads to a reference genome? So these are the things that we will cover quickly. Somebody is saying that they can't hear me. Can everybody hear me? Everybody except for Dr. Raichaduri? Okay, if you, Sahil, can you hear me?
Yep, I can hear you.
Okay. Okay, so if there are some technical issues, again, this recording will be made available. And if you want to work through, Sahil can probably help you see where the settings are for the audio. Okay, so this is what we did last time. We did pre-processing and then we did mapping. And so pre-processing is related to the way that the data is generated. The data is generated by taking the raw sample that you have, extracting DNA or RNA, and then converting it into short fragments by sharing the DNA or RNA. And then you have these short sequences of about 200 to 300 base pairs long that are sequenced. And the process of sequencing includes having this PCR amplification, bridge amplification, and adapters that are connected to the beginning and the end of these portions. So as a result, the output of this process are these FASTQ files. And the FASTQ files could be single end or pair end. And here you can see the example of pair end, approximately 250 base pairs long. And for each nucleotide, for each position, you have a quality score as well as the sequence itself. So that's the important thing to remember about this process. There's a number of technical things that you have to remember about how the data was generated. And then we mapped these reads onto the reference genome. And when we mapped them on the reference genome, here we saw that this was the reference genome. It has a sequence. The full sequence could be individual chromosomes or could be the full genome. If it's, in this case, we looked at a viral genome. And we see abundance of reads, like this area right here, that shows a level of expression. So we are looking at mRNA. So we are looking at a level of expression. And how do we detect that region that has some abundance? By seeing it visually like this, for example. So what exactly are we looking at? We're looking at genes that are encoded in the reference genome. Some of those genes are transcribed into mRNA. And the mRNA is in transcript format. That means that you have some exons are connected together by removing the introns. And then that mRNA is shattered and PCR amplification and adapter ligation is applied so that it could be read in the sequencer, right? So this is the raw data that we are dealing with. So now that we have this raw data, what we need to do are 3 basic steps that are involved in the processing of next generation RNA-seq data. These are the 3 major steps. And then later on, after I'm done, Sahil will go into more detail about what these steps are, just to kind of refresh everybody's memory. So we do pre-processing, we do mapping, and then today what we're talking about is quantification. So quantification means how do you count? How do you count and what are you counting? Okay, so the way that you do this and the way that we did it last time is we built a pipeline. Okay, so the pipeline that we did, there were 2 pipelines that we did. The first pipeline was simple. We took the pre-processing steps, as you can see right here. Then we did mapping on transcripts. These are known transcripts that are recorded in the reference file. And then we did RSEM quantification. We also did another pipeline that was expanded. And again, don't worry if this sounds a little bit too fast right now. Sahil will go over and we'll be able to answer any of the questions that you might have on these individual steps. So we did pre-processing here. Then we did an expanded mapping on genome and junctions, which are the connections between exons. And then once isoforms were constructed, again, we did quantification. So these two pipelines are examples of how the whole process, all of those three steps, are connected into a single process. This process takes the raw FASTQ files, kind of removes some of the elements, some of the sequence elements from the raw files, and then those files are mapped onto the reference genome, and then we want to count, what did we get? So today what we want to talk about is what did we get, right? And so what we'll do is we'll go into the pipeline. This is my pipeline that I ran last time, but each one of you should have a pipeline that you've run in the session that we did last time. So let's go over to the server, and I'll show you how to find the pipeline that you ran last time. When you log in to the server platform, you should see a list of pipelines that we ran last time. Okay, so if you ran multiple pipelines, for example, you did the online sessions, you will see them and you can see when this was ran. So this was run on the 21st of March. So this is what I did last time, and you can see that these are done. They are completed. For those of you that are having trouble with the audio, you can also call in using your phone. In the invitation, there's a phone link that you can just pick up your phone and call in. So these are the two pipelines that we ran last time. And I'll just take a look at the last one, so the one using HiSat2. Okay, and when I open, first of all, it should say 100% done. And then you should see all of the steps that we just looked at. And right here, you will see 3 folders. And in this folder, download pipeline output files, this is where we want to get to. Okay, so these are some of the files. So what you see here are the mapping statistics. So this is statistics on how many reads mapped onto the reference genome. You can see all of the results that are just zipped into a single folder if you want to download all of them. But then here really is what we're looking at. We have expression of genes in FPKM and expression of isoforms in FPKM. What is the difference between genes and isoforms? Genes are the sequences that are typically recognized as a consensus of a number of different transcripts. Transcripts are the actual variations of those gene sequences that we find in the expression table. So typically they're called isoforms or transcripts because they have individual exons spliced together. And this is why I say typically, is because sometimes they are not always, the introns are not always excluded. Sometimes they also have some introns present, especially in different diseases like cancer. But here, we're not going to look at the mapping statistics, and I'll just briefly explain why. The PDX project that you're working with was taken from the full data set and reduced to a smaller number of reads so that these kinds of pipelines don't take a long time. Today, we'll take a look at that expression table, but also we'll take a look at a full expression table from a different project, just so that you will know. So what are we going to look at? Let's take a look at the expression genes FPKM. So you will see that for each one, for the genes and for the isoforms, you have not filtered, which means that the genes that have 0 expression across all samples are not removed. Then you have those that are removed in FPKM. And so that is what we want to get. So let's download that file. And let's take a look at what this file has inside. Okay, so this file contains a number of columns. You can kind of make them a little bit bigger. And in the first column, you have a long list of rows. In fact, you can see that this is okay, 30 or 3,625. So as I said, the number of reads was reduced. As a result, you don't have as many genes as you would typically expect. And what kinds of genes do we have? We have ENSG, which is ensemble gene ID for human genes. You will also find in here ANS-MUS, for example, right here. And that stands for ensemble mus musculus, which is the mouse, gene. So these are mouse genes. And then you have XLOCs. And XLOC means that this is an unidentified gene. Again, because we reduce the numbers of reads, you have all kinds of sequences combined from those reads that are unidentified. So I hope that this kind of clarifies what is this table. This is called a table of expression. And here you have the samples. You can see that the samples are annotated. You have ER, which stands for ER positive, and TN, which stands for triple negative. So let's take a look at some of these things. What are XLOCs? Just again, XLOCs are unidentified sequences. They were not found in the GTF file, but cufflinks that we used here still assembled them into some kind of a sequence that looks like a gene. In this specific project, the reason why you're seeing a lot of XLOCs is because the reads were artificially prepared so that the sequencing, the processing time of the reads takes a smaller time. So as you can see, we downloaded this file, and this file is an FPKM. What is FPKM? So FPKM is fragments per kilobase per million of mapped reads. In other words, this is the number of reads mapped on the gene divided by the total number of mapped reads in a given sample in millions, multiplied by the gene length in kilobases. Now the formula is actually very simple. All it says is that because these are different samples that were maybe prepared in a different way, they have a different number of total reads available to that sample, and the genes are different. Some genes are just 1,000 nucleotides long, some of them are maybe several thousand nucleotides long. So just on average, if you have those two variables, you might want to account for that variation when you compare a group of samples versus another group of samples. Why do I say a group of samples? Because in this case, we have a number of representatives under triple negative and a number of representatives under ER positives. So what we would call biological replicates. Because of that, we want to kind of say that actually we want them to represent the same thing. We're really trying to study the ER positive condition and the triple negative condition, or the different mouse models that we have here. Okay, so this is the strategy that we're using, but you have to be aware that other strategies exist as well. You can get the raw counts, and you can also get TPM, transcript per million. We'll not talk about them now, but just so that you're aware. So as a result, what we see is that the column names are samples, the row names are genes, and in here in the middle, we have these numbers that are in FPKM. FPKM, fragment per kilobase per million. How do we visualize this data? So now we have some gene expression. How do we visualize gene expression? Let's take a look. So first of all, we did share with you in the zip file an Excel sheet that gives you some of these examples. But we can also take a look and just select one of the files, or one of the genes, sorry. So if I just select any kind of a gene, just some random one, ENSG, whatever, 10610, I can just insert a bar plot to see what is its expression. So what you see here is the height of the bar is going to be the level of expression. And just so that I can see what are those samples, I'm going to annotate them. So I'm just going to select this top column because these are the names of the samples. So you can see that I have each sample, and for each sample, I have some kind of a level of expression. Okay, so if you just quickly look at this, on average, okay, just by looking at it, and this isn't some kind of scientific method, but just by looking at it, you can see that the triple negative that start from right here are more highly expressed, right? If I take all of these and I add them together and I divide them by the number of samples I have in this group, they seem to have a higher expression than here. Okay. So this is an example with EN as G. I have other examples as well. I can add here another gene. For example, I can take this gene right here and see if it has a different profile of expression. So this was, sorry, let me forgot which one I got. Take this one. And I just use this as the y-axis. Okay, so here's another gene. The orange is another gene, and this gene has a different profile of expression. So now we have these thousands of genes that all have different profiles of expression. What can we do with this information, and how can we analyze it? One way to summarize our data, instead of looking at individual genes, we can look at the sample as a whole. And we want to evaluate whether the sample as a whole can be compared. So one way to visualize is using box plots. Sahil will go through this in more detail. I'll just quickly explain what are box plots and why are we using them. So box plots show us where is the majority of the data, right? The majority of the data, for example, in this sample, in this condition, is within this box. The box shows 75% of the data. And the middle of the data is the median. And so you can see how you can use box plots to compare conditions, for example. If we do box plots for this chart, it will look like this. Why is it looking like that? This is kind of different from this. The reason is because all of our numbers are very small, and we only have some outliers that are very high in their expression values. Because of this, all kinds of methods that are going to be sensitive to the variance of the data are going to be a little bit less sensitive because the majority of our data is way down here and is not fully represented. To address this, we're going to take a different data set and we'll walk through how to adjust this problem. This is a common problem with next generation sequencing. A lot of the values are very small, but some are very high. Okay, so the way to do this, again, this file is in the Dropbox folder that was shared with you. We will take this, and this is again two conditions. This is cloud and low and normal-like. The green are normal-like, and these are cloud and low. And so if you just take this table and you create a box plot. Sorry, let me just skip through some of these slides. If you create a box plot, it will look like this. So the box plot of this new data set, exactly like we saw in our PDX file, everything is down here and we can't really tell what is going on. The majority of the values are underrepresented. If we transform our data into logarithmic scale, so this is now transformed to logarithmic scale, we will start seeing a lot of our data in a much clearer way. Okay, so that is one step that we want to do. Transform our raw signal to logarithmic scale. After we do this, okay, we can now start visualizing the variance and understanding the difference between the different conditions that we have in terms of variance. To do this, we will create a pipeline that will do both things. It will normalize the data, and then will give us a visualization output that we can study in terms of our samples. So Sahil will explain more what is PCA, but for now, let's go to areas of analysis. So you should see this full overview of the types of data that you can analyze. And you will notice that on this side, under data mining, you have this section called utilities. Utilities are different scripts that are going to be useful for this kind of data transformation. And so I want to point your attention to one of these methods that we're going to be using today. So let's go under utilities and upload the data. So that PDX gene expression table, which should have something like FPKM at the end. If you upload this data, press on continue and start. And let's open this quantile normalization. So now that we've talked about the box plots, you can see that transformation of our data set is going to help us compare the different conditions in a more accurate way. Because our conditions are represented by a number of individual samples, we want to kind of normalize that variance within each group. So if you look at here, you can see that the median and the distribution of samples are different, and we want to align them. We're not changing the individual values. We're going to take the noise and kind of separate it from the signal by normalizing it according to median and to the variance of the samples. And as a result, if you compare two replicates, biological replicates, they should look a lot closer to this than if you were comparing raw samples. So let's first of all transform it to normal logarithm. Right here, you can see that this dropdown allows you to do it without manually converting your table. The second thing that you will see is a threshold. So let's take a look at what is the right threshold for what we are trying to do. Now remember, this is going to be a threshold in the logarithmic scale. So let me open this Excel that we already prepared so that we can take a look at that data. So this was our original expression table. Here we converted it manually to logarithmic scale. How did we do it? We just took the original value, logarithm, and +1. Why? Because logarithm of 1 is 0, so actually it doesn't change anything. So we did this transformation manually so that we can create a box plot for our data. Okay, and like you see, still this box plot is not very good because this specific chart was prepared artificially, because we selected certain reads to include or not to include in our main data. But what you can see is that it's still better than the raw box plots, right? Here you have a lot of outliers and the majority of your data is inside. So if you look at the second, just to go over the second example, the second example that we provided with the cell lines, you will see a very similar picture. Let's take a look at that really quickly and see what we can see in that example. Let me close this really quickly. This is, sorry. Yep, this one. So here we essentially did the exact same thing. We took our raw data, expression table, from the same pipeline that we did, converted it to log scale. You can see that this is logarithmic scale of this value. And now we can compare, this is the raw data. You can see that all of our data is at the bottom. And this is what logarithmic scale does to it. So just by looking at these numbers on the side, now these numbers are in logarithmic scale, natural logarithm scale, we can see that the majority of our data is under 5. So what we want to do is we want to leave this alone, okay? We don't want to normalize this. We don't want to adjust it. What we want to do is we want to take everything else. And so back on our threshold, we want to put in the number 5. And when you work through a lot of these next generation sequencing data set, what you will find is that a lot of the expression data fits this threshold. And you can study it more by looking at summary statistics and seeing the distribution of your data, that the signal is underneath this threshold of 5 in logarithmic scale. So now we adjusted our raw data. And the second thing, to visualize the samples, we want to use the principal component analysis. That also will help us visualize how these samples are distributed in this space of all the genes. So here, just make sure that you don't need to use a filter, and we do not need to transform it again to logarithmic scale. So we're going to just say no right here. And you don't have to worry about the other settings here. Click on save and run this pipeline. Okay, so now we have to go through what are we doing to visualize these samples. We need to explain what is PCA, what is principal component. Sahil, I'll turn it over to you. Maybe there are questions that people have, so maybe address the questions first, and then we can go through the PCA.
Sure. Thank you, Elia. And hello, everyone. I do see that there were some questions, and the first question that came was from Ramesh, that why do we use the why do we transform to logarithmic scale? So let me share my screen. Okay. So as Ilya was discussing about it, that since since that with the raw data we don't have like a very good result, we just have the outliers, and we don't actually see like what's there in the data. So in order to get a clear understanding of what's there in the data, we convert the raw data to the natural log, and then when we do the box plot, we do see that the data is better. We could see what's exactly is there. We could see the median, and that's how we could analyze the data in a better way. So that is the reason why we transform it to the logarithmic scale. And okay, so and please do let me know if there are some other questions as well, or if I would have missed out something. So please unmute yourself and do let me know. I would like to start from the very starting. Okay, yeah. T, do you
Hello,
wanna?
I'm sorry, Sahil. I just have a question.
Sure.
I'm myself from Asia, and I just have a basic question. How do you map a reference genome? As you see, like for example, in the NCBI database, like you have different versions of the genome. For example, GCH 37, 38, and how and to which one you actually map, and how do we know, like for example, the obtained data that is mapped to 37 or 36 or the earlier versions? Yeah.
Okay, so like while we run the pipeline, so over there we choose the latest assembly, that is the CH38, right? So we map it to the genome using coupling or cuff merge, or if we have to map it to the transcriptome, we use bow tie 2T. So I mean that's how we map it, and we can track down at each and every step what exactly did we use for the reference genome. So let me switch on to the server. Just a second.
Okay. And can we see a difference between like different versions? Like when you map the genome, they have like different assembly, or the assembly will be remain the same?
I mean, there would have been some changes in the assembly, so you would definitely look for some differences in those assemblies. But I won't say like they would be like 100% there would be some difference in this. There might be some minor differences, which might not be covered in the previous assemblies. So those are some things that might be added to those new assembly. But other than that, more or less, it's the same thing.
Okay.
I mean, yeah, but I can get you a more definite answer about it, because
Mm-hmm.
that's as per my understanding. But I mean, I can email you soon, like if I when I discuss with the researchers and the faculties about the same thing, like what's the difference
Mm-hmm.
between the previous assemblies and the latest assemblies, so I can email you that answer as well.
Okay, I have one more question.
Sure.
As Elia Brodsky was mentioning about the different isoforms
Mm-hmm.
that exist, like, and we can identify them depending on the abundance. Okay,
Yeah.
and if we go to NCBI database, and if you see, for example, from your RNA-seq data, there is a
Mm-hmm.
gene that has a differential expression. Okay,
Mm-hmm. Mm-hmm.
and this gene, when we go and check in the NCBI database, it has like different isoforms. Even there are like a lot of predicted structures with NM.
Mm-hmm.
So how do we identify that the data that we obtain from the RNA-seq and the isoform that we see in the NCBI database that
Mm-hmm.
matches exactly which one? And even the isoforms, they have like different sizes. I mean, like they may vary, like they have like different exons. Maybe one exon is skipped or one exon is added, or you have more intron sequences.
Mm-hmm.
So I just want to like know like how you can identify isoform that you obtain from the raw data, and how can you identify them in the NCBI database?
Okay, well, that's a good question, and unfortunately, I'm not very sure on answering this up. So I did make a note of this question, that how exactly we're gonna find out like what exon did we get and how we're gonna compare with the NCBI one. I would have to email you the answer for this as well, because I'm not sure on this one.
Okay.
Yeah. Uh, yeah, I'm sorry about that, but I'll get back to you soon after the call. I'll email it to you.
Yeah, sure, for sure. Thanks.
Sure, I'm sorry about it. Okay, I have something else. Okay, yeah, Akhil, I would be emailing it to everyone. Uh, okay, is it T? Is it in all cases the data must be transformed to log scale manually? Which function on the platform will do that? Okay, so I'm not sure who is T exactly, but T, we do have the functionality to do that thing on the server as well. So as Ilya was walking us through the pipeline that says the quantile normalization. So when you go to under utilities, and when you do the quantile normalization, there's an option that says just a second. Let me open that pop-up. There's an option that says transform table to log to natural log. So that's how we can do it via server or the platform, and we can do it manually by just selecting the function on Excel saying that says ln, and you can do that thing manually. So that's how you do it manually and on the server. I hope that answered your question, T. uh, is there any other questions before we actually move forward and I go ahead and cover the things that Ilya did in more details? Okay, so I'm hoping that there are no questions, or there is one. What are the settings for PCA? It looks different now. Okay. Okay. Is it that is because yeah. Okay. That is because you are doing quantile normalization and then the PCA directly. So that is why there would have been some differences in the settings. But if you are just doing the PCA, so the settings would be the same that you would have seen in transcriptomics one. Can you show the pipeline flow up to today lecture? So which pipeline do you want to see, Harshada? Like, I mean, the RNA-seq pipeline or the PCA pipeline? Mm, The second one, that is a PCA. Okay. Okay, sure. Yeah, I can. Yeah, I can do that. So that's the login screen of my platform. I'll go under utilities. So over here, I have an option to upload my files. So I'll click on that. I click on add files. Now everyone was emailed the zip file, and I mean there is a link to the files on the discussion board as well on the home organization page. So you can take the file from there, and then we'll use the cell lines expression gene keep fpkm filter 6. That's the one that we're going to use for our PCA. So we're going to just upload it, and I could see that it's 100%, so the file is uploaded, and I'll click on continue. Now I have the start button, so I'll click on start. Now the first thing that I'm going to do is the quantile normalization, so that we remove any noises or any discrepancies in the data, and make sure that the data is normalized and it's clean. So I'm going to go on quantile normalization. Now it's important that we convert our raw data to the natural log. So I'm going to say it as yes, and we can have different threshold value. Either we could have it 0.0. So the only thing that if we have 0.0 is that it will normalize each and every sample, and which might take a longer time, and which might not be very useful as well. Ramesh, just give me a minute. I'll just finish this up, and
Yes,
then I'll
yes, yes.
complete.
Please, please,
Yeah.
please. Go ahead.
So over here, I'll go ahead and select a random threshold value. But this random is just because, I am running it as an example. But when you do it as a part of a project, or if you do it as a part of a course, so then there are some specifics that you have to keep in mind while selecting the threshold value. So I'll just go ahead and do it as 5, and I'm gonna go and click on save. Now the option that is left to do is the PCA. PCA is principal component analysis. I'll cover like what exactly is PCA and why do we do the PCA, what's the importance of the PCA. I'll cover that thing in some time. So over here I have my default factors, and I have already converted it to the log scale in the quantile normalization. So I won't do it yes over here. So I'll do it no, and I'll keep all my factors the same and continue for now. So I'll just go and click on save, and after that I'll go and click on end, and then it's asked me to name the file, name the pipeline. So I'm gonna just say PCA test, for example, save, and then I click on run pipeline. So that's the PCA pipeline that we were supposed to run. Okay, I have some questions over here. Okay, Ramesh, do you wanna
Yeah, actually, it's like regarding the PC, as you said, like you're going to explain in some time. It's okay because I was like curious to know like why you have like different quartiles, like quartile 1, quartile 2, quartile 3, quartile 4.
Sure.
Or else, like, does it have some kind of difference when you have like two different quartiles? For example, quartile 1 and quartile 2, just not like having four differences. But it's okay, as you said, you're going to explain. I will just wait for some more time. Thank you.
Okay, sure. Thank you, Ramesh. Okay, so I see the question from Titloop. If my data does not have gene expression data, what step should I start from? Mapping, or will PCA show me anything? Okay, okay, that I mean, if you're gonna do the RNA-seq pipeline, if you're gonna do RSAM expression table, so there would be for sure there would be some kind of an expression, because I mean if there is a zero expression, so then I mean that doesn't make sense to do the studies then. So I mean there might be low expression, but there would be some kind of an expression. And just to verify that whether you have an expression or not, that is why when you do the PCA, because PCA gives you a pictorial representation of these samples in a 2D plot. So I hope like this answers the question. But I mean there would be like a very rare case that you're doing some kind of a study and there is not at all any expression into this data. So there would be something, it might be low. Sayak, I would suggest you to get in touch after the class so that I could show you what exactly did we do in the previous class. For now I'll just continue with the lecture for today. So Sai, please reach out to us later. Okay, what settings for the PCA, Nathan? So for the PCA, we just make sure that we don't have we are not converting the log scale factor as yes, because we are already doing that in the quantile normalization. And for the lectures in transcriptomics 1 and 2, we are keeping the PCA factors as default. The settings are as default, but but you're gonna get a chance to play with those settings when once you're gonna start working on your own project. So that's when, like, you're gonna understand, like, okay, this is something that you have to change according to the data, or this is what you are required to do that during that project work. So that's when you're gonna change the settings for the PCA. I hope I answered your question. If not, please feel free to unmute yourself or type down in the chat box, Nathan. Okay, Chita, by normalization, we are removing the outliers. What will happen if those are the important one? Okay, yeah, that's really good question. I mean, we are removing the outliers primarily because so as to see, like, what the data looks like. Because if we just work with the outliers, we are not gonna get to the analysis of the real data or the actual data. So first, we're gonna remove the outliers. We're gonna work with the actual data so that we get a feel, like, what exactly the data is and how does it look. And once you get an understanding of the data, then it might make a lot of sense to go and work with the outliers if at all they are important. But just working on the outliers and leaving the actual data won't make any sense or won't lead to any better analysis. I hope I answered your question, Chita. If there is something, please let me know. Yes, Soham, the recording for the today's class will be provided as usual via email, and it would be uploaded on the organization page under the forums as well. Okay, what will be the first step after having the raw RNA-seq data from a fresh biological sample? Okay, so that's what that was it. That is what we are doing today, Harshita. That in the last class we had an data raw data, and then we uploaded it. We did run a RNA-seq pipeline, and then after that we got the expression table. So that's what we are doing today, that we are going to quantify, or we're going to analyze that expression table. We're going to download it. We're going to see the expression values, like how Ilya was mentioning it today in the morning, and was mentioning that we're going to plot the bar graph just to see the expressions in different samples. Like, let's say if it's a TN triple negative, or if it's a ER positive sample, so what's the expression values. And then after, once you're clear with the expression levels and the expression values, you're going to move forward. You're going to clean the data, and then after that you're going to run the PCA so that you could see the visualize the data. So that's still today. And after that, like, what exactly do you do after the PCA, that will be covered in the next class. So there's a whole process that you do once you get the raw sample. So for more information, like, what exactly do we do with all those kind of a data, it's better that if you could go back to the online modules and go through transcriptomics 1, 2, and 3. So that would give you all the steps that are required from, like, getting the biological data or like a raw data till the point where you can derive some biological interpretations. Okay, Harshita, I hope I answered your question, but still, if there is something, you can unmute yourself or you can type down in the chat box. Smith, I'm gonna email you the presentation, which explains clearly, like a pictorial representation of, like, what's the difference between expression genes and expression isoforms. So I'm gonna email this answer to everyone as well. Okay, is there anything else which you guys want me to go through before I actually jump on? Okay, and Mohit, I hope you are okay. One replication is sufficient. One replication for? Sorry, I didn't get your question, Harshita. Okay, in the meanwhile, Mohit, it would be nice that if you could note down all the questions that are there in the chat box, so that we could put up on the forum and we could get the answers for everyone.
Sure, Sahil, sure.
Yeah, thanks, Mohit. One. Okay, okay, one replication is sufficient for biological sample. Harshada, do you want to unmute yourself and explain me like what exactly do you want to ask? Or if you could elaborate the question exactly, like for a biological sample, what replication do you want to ask about? Okay, in the meanwhile, I'll go to Prabhuda. Can that logical cookbook style command table for okay, can there be logical cookbook style command table for both the pipelines? Uh, for what both the pipelines? I mean, do you mean the RNA-seq pipeline and the PCA pipeline, Prabhuda? Okay, yep, there could be like a cookbook. Okay. Yeah, sorry. Just give me a minute, Chita. Yeah. So Prabhupada, there could be there is a cookbook style command table for both the pipeline, and I can share that with you via email. Like how does that exactly looks like? Okay. And minimum how many biological replicates are required for the analysis? So it's better like if we are doing the analysis, so at least have two, so that we could have some kind of a comparison that could be done. So I mean, if there is just one apple, so you're not going to compare it with any other fruit, right? So it's better that if you have one apple and one orange, so that at least you could compare their features, you could compare their size, colors, and etc. So it's better to have minimum two, and you can go anything to maximum. Okay, I hope that answers your question, Chita. Okay, perfect. Okay, so I guess all the questions are being answered, and Mohit is noting down all the questions that were there on the chat. So we're gonna create the forum for all these questions with all the answers to them. So you could review it, and if there is something, you can comment into that forum. So like it could be more like a healthy discussion over there with all the participants participating in that question answer round as well. And again, feel free to like stop me, or if there is something. In the meanwhile, I'll just go and start with the other things. So one thing that I wanted to go through before we actually jump up into technical things for today is that I have been getting a lot of questions from the students or from the participants that they are having trouble in going to educational site and then enrolling for a course. So let me cover that aspect first before I move forward. So that's the URL for our educational website, edu.t-bio.info. Everyone should have their credentials and they should be able to log in. Since I'm logging over here, I could see that like there is a name and then there is a profile pic over here. So that is the sign, or that's the identification where you can see that you're logged into it. If you're not logged in, there will be an option to sign in or to register. I mean, the login or to register. So that's how you're going to log in or register. Then after that, there is a section that says courses. So over here, there's a drop down. You could see that there's introduction to bioinformatics, transcriptomics 1, 2, and 3. So let me go to transcriptomics 1 just to see like how does the course exactly work. The other important thing that you should or you might should know about it is like if you're doing lecture 1.1, let's say, and then you got bored, or if something comes up urgently, so you could leave that lecture then and there itself, and then you want to continue next time, so you could continue from the same point where you have left. So there is nothing like you have to complete all the lessons in one go. So there's nothing like that. So this is how the whole course looks like. So I'll go on first lecture, that is 1.1. So I can go through whatever it is and I can mark it as complete. So I'll read through this, what are the videos, what are the pictures and everything, and then after that there is an option that says mark it complete. So that's how like you could keep a track of the progress, like okay, this is what you have done last time, and then you have to start from lecture 1.2. So that these are some things that I've been getting a lot of questions about, so I thought like I'll cover it in the class with all the participants being on. So this is how you go to the lessons, this is how you access the lessons on edu site. And so like somebody asked me like what is the whole process like when you get a raw data to a point where you get a like biological interpretation. So over here like everything is clearly mentioned, like what are the logical steps for analyzing the RNA-seq data, how exactly do you do the analysis, and then what do you want to do with your gene expression table. So everything is clearly mentioned in the lessons that we are doing it in an online session. So that is why it's highly recommended that after the session you go back to online modules and go through them step by step and follow each steps. If there are questions, so you can ask in the next class, or you could email us, or you could type down in the chat box. We have a chat box over here. I saw that some of the participants are utilizing this chat box to answer any questions. But still, whatever way is comfortable for you guys, you can do that, email or chat or whatever. It's easier. So I'll just walk through the presentations again, just so that if Ilya might have skipped something, or if you were not sure like what did Ilya go through, so we can cover those things again. So this is what we covered in the last lecture, like how do we gonna process the NGS data, and like what's the role of pre-processing in the standard RNA-seq pipelines. So like as I was mentioning, like it's way too important to clean the data, to have a good data in order to get the expression table, and that is why we do go and do the pre-processing. And then what are the mapping techniques, like where to map it, like is it to be on the transcriptome, or is it on the genome, or if it's gonna be like a combined strategy for both of them. So that's what is gonna it's being covered in the mapping techniques. Both of these things are covered in transcriptomics 1 in detail, so kindly go ahead and look at the lecture what exactly it says. The other thing is like the shotgun sequencing. So this is the process that that's how the sequencing generates the reads, and these reads are short, so they are amplified using the PCR. And then after that we do use the adapters of a specific kind, and then these are used to identify the beginning and the end of the short read, and that is how we get our raw data, and we clean that particular data to get a good quality of a FASTQ file. So that's the whole thing, how exactly do you get the reads and how do you obtain the FASTQ files. So this was the result from the last lecture. This is the pipeline ID that you can go ahead, and if you want to download the results and want to interpret it. But just to quickly go through and glance through what was exactly it. So we took the reads and then we mapped it to the reference genome, and we do see that they are in abundance. Over here we see that there is a region, that's the gray region that I'm talking about. It shows the abundance of the reads that are mapped to that particular read. So that's how we're gonna say okay, if our reads are in abundance or if they are in less. So that's how we're gonna figure that out. This is all there in the recordings of the previous sections and in transcriptomics 1. This was something that Ilya have already mentioned, there are genes that are expressed and some of them are not expressed, and how exactly we do mention that they are expressed is they are converted into smaller transcripts, and then the transcripts are shattered into the reads. So that's the process, how we could say that okay, there is some kind of an expression in the genes and there is no expression with the genes. So the 3 steps that we do follow, and 2 of those are already done in the previous lectures. The first one is the pre-processing and the other one is the mapping. So now today we are going to go and do the quantification of the expression levels, that how exactly you're going to quantify those expression levels and count the expression levels. So these were the 2 pipelines that we did last time. So we first of all we mapped it on the transcriptome. That is why we used bowtie 2T, and the other pipeline was mapping it on a genome, and that is the reason why we use cuff links and cuff merge. So I'm hoping everyone would have got these expression files and they might have run the pipelines. If not, please go ahead and do run the pipeline so that you actually follow up what exactly is happening in the lecture. But for now I'll just go to the same pipeline that was done last time. So this was the pipeline that was done last time, and I could see that it's done, it's completed. Over here it says 100% done, and now I'm gonna go and click on download output file. I could see it's exact same thing that I'm seeing on my presentation. So for today we're gonna work with expression genes fpkm.txt. There are different other output files as well, fpkm not filtered and isoforms file. So those are different output files that we do get. Non-filtered ones are the ones in which we're gonna get the genes that are not expressed as well. So you're gonna see like a lot of zeros or like a lot of junk values in non-filtered. But in expression genes, FPKM, we're gonna get the genes only for which there is some kind of an expression. There might be like zero expression as well, but you're not gonna get any junk values in the filtered, in the FPKM filtered file. So okay, so I'm gonna open my filtered file. So as it was discussed today that over here we have all our genes, and these are the samples. So ENSG, that is something that um, that comes from the GTF file. I'm gonna tell you like what exactly is the GTF file. And so I mean there are, so this particular expression table that we got has the combination of the mouse gene and the human gene. So ENSG is something that represents the human gene, and ENS muse is something that is the mouse gene. And there could be like XLOC. So this is something that is an unidentified transcript. So we don't have to worry about an unidentified transcript for our analysis, but we do take care of the human ones and with the mouse, right? So what exactly are we going to do or quantify the expression? Like, what exactly are we going to do of this Excel file? I mean, this is something that, doesn't make sense because it's all numbers. So what we're going to do is, like, let's say we're going to select this particular gene with the samples, and I'm going to plot over here. Okay, so this is a box plot that I did get just for a particular gene that I was working with. But now over here on the y-axis, it's just the numbers, so it doesn't make sense, like, what exactly it is. I know that this is an expression value, but what is this? So for that, we're gonna go and do the select data. I'm gonna do this edit, and just a second. Okay, so now I have my samples over here. Okay, just a second, something happened to my Excel. Okay, so then I have my samples with me over here on the y-axis. So what exactly do we see in this particular graph? Like, do anyone want to answer this thing? Like, what do we see exactly over here? Or like, what exactly could you interpret from this graph? Anyone? Okay, I'll give an answer. Increase expression in TN samples. Okay, that's great. Yeah, that's exactly the thing, Vogue. So we could see that there is an expression level in the triple negative samples, but there is like no expression or very less expression in ER positive samples. So this is something like, we could see in via graph, by the box plot that we have plotted. So this gives us an insight. Okay, like, for this particular gene that we're talking about, ENSG, that's a human gene, and this particular gene does not have any expression or very less expression in ER positive, but has a high expression on the triple negative samples. So that is something to, like, worthy point noting. So we're gonna go and do some more research, like what exactly this gene does and why it is, like, it's down regulated in ER positive and high and upregulated in triple negative. So this is something that, that's included in the downstream analysis. So that is how, like, we quantify and we try to understand, like, how does the data looks like. So I'm gonna go back to my slides and I'll continue. Okay. So, and then we discussed about the standard measures that were used for the RNA quantification. So we do have FPKM, that's fragment per kilobase per million mapped reads. And over here we do have the formula that number of reads mapped on the gene divided by total number of mapped reads in millions, and then the gene length in kilobases. So just to keep the standard scale of metrics, we have made sure that all the units are into the millions, because I mean the genes could vary from anything that is from 1,000 to 10,000, and I mean there could be more genes, could be less genes, and for the larger genes the coverage is small. So that is why we made sure that the unit is same all over and it's in millions. So this is what we discussed. Like, this is how the gene expression table looks like. On the columns are over here with all genes, rows are all your samples, and these are the expression values that you get. And this is like a comparison graph that we had, like how exactly you could figure out like how the expression values look like and what's the difference, and how you're gonna interpret that particular box plot. So you're gonna do that. Now, how do we get the value of or like the sections like ESN, ENSG, ENSMUSG? So how do you get these particular genes into your expression table? So we do get that through the GTF file. So what does GTF file contains? It contains the beginning and the end position, and what is there down the level of the exon. So that is how we do get our information about the genes, what are there in our expression table. So this was just the overview of the GTF file, and then we were discussing about the box plot. And Ilya did mention about why do we do the box plot with the raw data, why do we do it with the logarithmic scale. So I'll just go over it quickly. So this is our table of expression. This is for the cell lines. This is not the PDX one. This is a different cell line, and this particular file is there in the zip file as well. So we have raw data over here on this side, and then we have the log scale over here. So if I go ahead and look in the formula, I could see that it's the natural log of that particular value, and then the +1. So the reason why did we do the +1 is the log scale of 0 is something that we cannot do. So in order to stabilize the value, we do +1 over there, and this and adding this +1 to this particular formula won't make a lot of difference, because anyhow the natural log of 1 is 0. If you want to confirm that what's the natural log of 1, so it's 0. So I mean it's not going to make any effect if we're going to do +1. So but just to stabilize the value and we don't get anything that is 0 or that is something not useful, a junk value, so that's why we just do +1 over there. And while we do the box plot, if this is the box plot of raw data, and over here we could see that we just have the outliers, and we don't see what's actually in there in the data, because I mean it's all merged in. And the signals are very low in this, because I mean we don't see the median. The median actually is like something that's there inside in very bottom, so we don't see that. So the analysis won't be that good, and it would be quite less sensitive. So that is the reason why we came up with a way of having like a box plot with the logarithmic scale, and we do with the natural log. And if after doing the natural log, if I could see the box plot, I could see that okay, my median is like over here. I could see what exactly is there in the data, what's the expression of the data, like what's the expression value of the data. So I could see those information. So now my analysis would be easier, and it would make a lot of sense, and the sensitivity to the data would be better in comparison to what we had in our raw data. So moving forward, So it's the same thing that I showed you on the Excel file. We have the raw data over here, and then we have the log value. We have the Claudin samples over here, normal like. So we have two different groups. That's what we are comparing with. So they are marked with different colors, and that's the log formula that we are using in our log scale. The files are there in the zip file, so you could go and it could explore more like what exactly the file contains. But now if you want to dig more into like the actual data and like the statistics of the data, so it's better that you do the descriptive statistics. So that's where you're gonna get some more information about like what's the overall median, what's the skewness of the data, what's the range, what's the minimum and maximum. So that would give you a more detailed knowledge about your own data set. So like like what's the minimum expression value in your data, what's the maximum expression in your data. So like to get a more good understanding about the data, you can do summary statistics and not just like plotting the box plot. The box plot gives you the information about the medium, about the first quartile, second and third quartile, but like it won't be, you won't be able to figure out all the information until unless you don't do the descriptive statistics. So yeah, if you want to have more information about it, so you could go dig deeper into it to find out what's your data looks like. This is just like the visual summary. Like if you do the descriptive statistics, like if you want to compare the median, like what's the median of your raw data. So the green one over here represents the raw data, and the blue one are the log scale values, and the red ones are the quantile normalized values. So you could compare the different medians in all the three forms of the data, or you could have the skewness comparison or whatever comparison you want to see for your own data set, so that you have a better understanding about your data. So that is about like like why do we do the log scale, what's the box plot, how do we do that, and what information do we get from the box plot. So this is again the similar thing, like this is for the raw data and this is for the log scale value, and what's the difference, where are the medians, where are the skewness and everything. So you could figure that out with this graph. Now the next thing that I'm going to cover very quickly over here, as we are running out of time, is about the PCA. Okay, so what is PCA actually? So PCA is principal component analysis. So let's say that you have gene 1, you have gene 2 with the samples over here, and then you have the expressions over here. So if you want to visualize your sample in a 2D space, so this is how it's going to look. You're going to have your X gene 1 on your X scale and gene 2 on your Y scale, and then all your samples over here. So this is good when you have just two genes, like gene 1 and gene 2. But if you have more than two genes, so then like let's say that in this particular example you have three genes, so then this becomes like a 3D picture. Or like if you have 10,000 genes, so that means it's 10,000 dimension. So PCA is a dimensional reduction technique, so that come whatever number of genes you have, you could convert them and plot it into a 2D scale and to a 2D graph, so that you could visualize the data, you could visualize the samples, like where exactly the samples lies and how does it look. So PCA is nothing but a dimensional reduction technique in order to visualize all your data. So when we do run a PCA, I'm gonna go to the platform to show like what output do we get and how do we do that. But when you do a PCA and you get an output file, it's an Excel file that you get an output, and over here you have different principal components, like you have PC1, you have PC2. So they have they range from like high variance to the low variance. So PC1 and PC2 have the highest variance, or like PC3 and PC4 might have lower variance in comparison to what there is there in PC1 and PC2. So when you plot PC1 and PC2, then that's where you get like you could get a clear understanding about the variance of your data. So that is when we do PCA, and that's how we analyze the PCA. So while okay, I got a chat over here. How many variances you can compare? So it depends upon like how many variances do you want to compare. Generally, like while we are doing the research, we just compare PC1 and PC2 because those are the two principal components with the highest variance. But I mean, like, if you find like there is something else important in PC3 and PC4, and you might get a more better information about it, so you could do PC3 and PC4 as well. So it depends more upon, like, what exactly are you trying to achieve through PCA. Yeah, Ramesh, do you have any question?
Yes. I just have one question regarding the PCA.
Sure.
So I just want to know, like, because now the data we use is like ER positive and the triple negative breast cancer. These are the submolecular types of the breast cancer. Okay, now you are comparing both the tumor types, but what happens like when you are going to compare with the normal counter tissue part of the tumor, and you have now like three differences: the normal tissue, and then the triple negative, and then the ER positive. So in this way, we can use the PCA to compare like the differences between the three, or it's only for two things. Like you can use only like two things to compare at the same time.
No, so PCA, I mean, you could just so PCA is nothing depending upon the comparison. It's just like, you want to see your data, you want to see your samples in a 2D space. So it just reduces the dimension. So if you have like three cases, the normal, ER positive, and triple negative, right? So it's gonna be like a 3D plot. So you cannot visualize a 3D plot, or you cannot extract any kind of information using the 3D plot. So that is why we reduce the dimensions to a 2D graph, and then we visualize, like, where do we have the groups, or where do we have the samples getting clogged. So that's when you do PCA.
No, I mean, like, for example, if you go to the PCA plot that you showed us before, in that you have like the differences like in the sample and the expression levels, right?
So this is a PCA plot that we
and you have the PCA values for these two genes. Okay.
Yes.
For example, let us consider for the RNA-seq data, it has like a lot of thousands and thousands of genes, and we have the information. But if you want to compare like between the molecular subtypes and as well as the normal tissue part, so how can you plot like this? Because now you only have two genes. So you use a PCA component and you plot them using the 2D plot. If there are more genes that are involved, so you go for 3D plot, or more than two, you use a 3D plot, right?
Yeah.
So this is why we use like PCA component 1 or component 2. But this is the case for just for two things. Like, but to compare like many things, and PCA can be used, I don't know, because I'm entirely new to this statistical field. I just want to know. Yeah,
sure. No, so that's what I'm saying. See, okay, there are two different things. One is to compare, right? So what you are trying to say is that you want to compare normal with ER positive and triple negative, right? That is the first thing. The second thing is, you're saying that, let's say that you have a study and that has 10,000 genes. That's the exact case. 10,000 genes means, like, you're gonna have 10,000 dimension plot. So see, over here, what I'm trying to say is, like, if it's to the gene n, right? So there would be a multi-dimensional space. So then how are you gonna visualize all the features in that 10,000 genes? So for that, you need to reduce the dimensionality to a 2D plot, so that you could visualize all your samples. Like, okay, sample 1 is your normal sample, sample 3 is your ER positive, sample 7 is triple negative, right? So this is how you could visualize in a 2D scale. And then after that, once you get that particular thing, once you get this a 2D graph, then following the downstream analysis, you could do further analysis, like, to compare, like, how does the normal respond, how does the triple negative respond to a particular drug, let's say. So then that's like a sub analysis that you're going to do after you achieve your PCA. Does that make sense?
Okay, thank you, Sahil.
Yeah, I'm going to share more documents on PCA. I can put it up on the forum so that you get a better understanding on this. Okay, so I guess we are running out of time over here. So just like a homework for you guys is, in the presentation there is a task that we did show how to run a PCA pipeline. So just go ahead and try PCA. There is more information about the PCA in transcriptomics 1, 2, and 3. So kindly go through it. In the meanwhile, I would share the documents and the answers to what Ramesh was asking about the PCA, and, like, we'll try to get a hang of what exactly was he trying to understand, and we'll post it on the forum so that everyone could see that particular thing. And I'll just quickly go through about the upcoming events. As everyone is aware that we have a neurobiology workshop coming up on April 5th. So I hope like most of you guys have registered to it, but if you haven't, please do that. It's an online free workshop for the participants of OmicsLogic training program. So please do that. If you don't have the link, I'm gonna send it in the recordings as well, so that you could go ahead and register it. And we are having our annual conference on computational biology and bioinformatics from April 5th to 6th. We would be the part of this conference, and we would be presenting our posters. So we would be sharing more details about the conference. And if you are in US, so you're more than welcome to be the part of this particular conference. Just reach out to us if there is any particular questions that you have regarding the conference. Before I stop talking about over here, I just want to make sure that Mohit, did you had a chance to note down all the questions that were asked in the chat box?
Yes, I have.
Okay, yeah. Perfect. Okay, before I end the call, Justin, do you want to say some words about what did we do today and something? Justin, are you there?
Oh, hi, Sahil.
Hi.
Yes.
Do you want to say something before we end the call?
Sure. So I found that very interesting and useful. So thanks for going through that. Hopefully all of the attendees benefited from that as well. So generally speaking, as a neurobiologist, and I know I made some of these contents last time, and I have made a number of your acquaintances in the past, but I'm very excited personally to be able, or for us to be able to give these presentations. And I think it's a really exciting time to be tying together computing and informatics and many different fields of inquiry. So thanks again to everybody, and I hope you share my excitement, and I hope we will meet virtually or in person down the road.